My lab has been interested in neurovirology and how peripheral inflammation can drive neuroinflammation in HIV infection. Monocytes in HIV infection are activated by interferon and LPS. Peripherally activated monocytes in HIV infection migrate into the brain and cause neuroinflammation and cognitive impairment. An increase in these activated monocyte macrophages can be seen in the brains of people who have died with HIV dementia or cognitive impairment. This has also been replicated in models of animals. Activated monocytes generate exosomes carrying significantly altered microRNA profiles compared to non-activated monocytes. The most highly activated mIR in monocyte exosomes activated by interferon and LPS was mIR 155. We hypothesized that an engineered exosome expressing an antagomir to 155 might decrease peripheral activation, neuroinflammation, and transmigration into the brain. We chose to use pluripotent stem cell-derived monocytes that would provide a uniform cell source with the ability to scale up as well as minimize human donor variations. The other advantage was that we suspected and confirmed in vitro that monocyte EVs would preferentially enter into other monocytes, which was our target. We grew up stem cell monocytes and then harvested the exosomes. We transfected an anti-mER 155 into stem cell exosomes and treated animals with these engineered exosomes, monocyte exosomes alone, and monocyte exosomes transfected with scrambled miRNA. Monocyte exosomes were characterized by size and tetraspanins and electron microscopy. We used a humanized mouse model or BLT mouse that's a good model for HIV studies. These mice were infected with HIV for 6 days and viral load checked. They were not treated with antiretrovirals. Engineered exosomes were injected at day 7 and 10 post-HIV infection. 2 weeks later, the mice were sacrificed and the blood was processed to evaluate inflammatory markers, HIV viral load, and spleen monocytes. The brains were processed for immunocytochemistry. The results show that the stem cell monocyte EVs alone decrease the percent of circulating CD14-16 positive monocytes, as well as the percent CCR5 on monocytes. Monocyte EVs also significantly decrease the percent CCR5 on CD4 and CD8 T cells compared to HIV positive non-treated animals. Anti-mER 155 EVs decreased the percent CCR5 on monocyte macrophages. When we stained for macrophages in the brain, we saw a trend toward decreasing macrophage numbers, suggesting a decrease in transmigration. In conclusion, we showed that EVs could be loaded with cargo, transported to target cells, and have biological effects in vivo, altering monocyte and T cell activation markers. These studies do not show an advantage in using the antagomir 155 to reduce inflammation below HIV infection alone, but the numbers of animals are small. There appears to be value in treatment with stem cell monocyte EVs in reducing CD16 positive monocytes.